In our previous video, we introduced the fundamental concept of DNA sequencing technologies, categorizing them into two primary types: short-read sequencing, also known as second-generation sequencing, and long-read sequencing or third-generation sequencing technologies. Both methods are shaping the modern genomics, with companies like Illumina continuously refining their devices to improve accuracy and speed. Today, we will focus more on the short-read sequencing, discussing its scientific principles, technological processes, and biological mechanisms that make it one of the most widely used sequencing methods in the world. First, let's talk about what the name short-read sequencing represents. So it basically refers to methods that generate relatively short DNA sequences with fragments ranging from 50 to 300 base pairs in length, since its introduction by companies like Illumina. This approach revolutionized genomics, making it possible to analyze large-scale genomic data at a fraction of time and cost compared to older methods like Sanger sequencing. Despite its widespread use, short-read sequencing does have limitations, particularly when analyzing repetitive or structurally complex regions of the genome. However, its ability to produce vast amounts of data quickly and cost-effectively ensures its place in genomic research, clinical applications, and even consumer genomics. Before we go any further and dive deeper into short-read sequencing and its real-life application, we encourage you to subscribe to our channel to keep up with our content, and to support the work we do, please hit the like button below. Short-read sequencing can be divided into 5 main steps, and in this video, we will focus our attention on the methods used by Illumina, as they are one of the leading providers of second-generation sequencing technologies. The first step in any short-read sequencing workflow is preparing the DNA sample. The quality and quantity of DNA are essential for ensuring high-quality results. The amount of DNA required depends on the specific sequencing platform, but typically, for whole exome or whole genome sequencing, anywhere between 100 to 1000 nanograms of high-quality DNA is necessary. If you have any question, or if you are simply curious about how DNA is extracted and purified, you can watch our video on DNA extraction. Okay, once the DNA is isolated, it must be fragmented into smaller pieces to fit the short-read sequencing platform's capabilities. DNA fragmentation can be achieved using two primary methods: sonication and nebulization. Sonication uses ultrasonic waves to mechanically shear the DNA into smaller pieces. The size of the fragments can be controlled by adjusting the strength and duration of the ultrasonic treatment. On the other hand, nebulization breaks DNA into fragments by forcing the DNA solution through a small nozzle under high pressure. Like sonication, the fragment size can be adjusted by controlling the pressure. In either method, the goal is to obtain DNA fragments within the desired range of 50 to 300 base pairs. However, during the fragmentation process, the DNA ends may become ragged or have overhangs, which must be enzymatically repaired into blunt ends for further processing. After fragmentation and end repair, a crucial modification is introduced, which is called A-tailing. This process involves adding a single adenine nucleotide to the 3' end of each DNA fragment. This modification ensures compatibility with the T-tailed adapters that are ligated to the fragments in the next step. These adapters serve multiple purposes, some of which are as follows. 1. They provide priming site for the amplification and sequencing steps. 2. They contain sequences that enable the fragments to bind to the sequencing platform's flow cell. And 3. Adapters may also contain certain barcodes that allow for simultaneous sequencing of multiple samples in a single run. This process is also known as multiplexing. Once adapters are ligated, some setups employ quantitative PCR or qPCR to validate the library's quality and ensure the adapters are properly attached. This step uses primers complementary to the adapter sequences, amplifying only those fragments that have been successfully attached to the adapters. In the library amplification step, depending on the sequencing application's requirement, the prepared library may undergo additional amplification to ensure there is enough DNA for sequencing. If the focus is on whole genome sequencing, the entire library may proceed to the next step. However, In whole exome sequencing or custom-designed targeted gene panels, only specific regions of the genome will be selected. To capture these regions, two main techniques are employed. 1. Hybridization capture. Probes complementary to the regions of interest are introduced to DNA fragments. These biotinylated probes bind to their target sequences, and the resulting complexes are captured using streptavidin-coated beads. After several rounds of washing, only the DNA region of interest remain, ready for either post-capture amplification or sequencing. 2. PCR amplification. Alternatively, primers designed to amplify specific regions of the genome can be used to enrich the target sequences. Repeated rounds of PCR selectively amplify the desired regions, ensuring that only the relevant fragments are sequenced. The next step includes cluster generation on the flow cell. With the prepared DNA library at hand, the next crucial step is generating a DNA cluster of each of the genomic fragments on the sequencing platform's flow cell. The flow cell is a glass slide coated with oligonucleotides that are complementary to the adapter sequences added earlier. The process begins with the binding of DNA fragments to the flow cell's surface. Then, a DNA polymerase enzyme synthesizes a complementary strand to each attached fragment, creating double-stranded DNA. The original strand is washed away, leaving only the newly synthesized strand bound to the flow cell. This remaining single strand then undergoes bridge amplification. During bridge amplification, the single-stranded fragment bends over to form a bridge, allowing its adapter to bind to a second oligonucleotide on the flow cell. A polymerase then synthesizes the complementary strand. This results in a new double-stranded DNA molecule, which is again denatured into two single strands. This process repeats itself until millions of identical copies or clusters of the original fragments are generated in each spot on the flow cell. The final step that we are going to discuss in this video is sequencing by synthesis. Once the clusters are generated, sequencing can begin using the sequencing by synthesis method. This technique involves the addition of fluorescently labeled nucleotides one at a time. Each nucleotide is tagged with a different fluorescent dye, so when it is added to the growing DNA strand, it emits a specific color of light. The sequencing platform records the emitted light, identifying the base at each position. Sequencing by synthesis is a cyclical process. In each cycle, a single nucleotide is added to the growing strand. The platform records the light emitted by the newly added nucleotide, and the cycle is repeated until the entire sequence is read base by base. This process generates millions of short reads, each representing a fragment of the original DNA. Importantly, sequencing by synthesis can be performed in both forward and reverse directions, enabling paired-end sequencing. In paired-end sequencing, both ends of a fragment are sequenced, doubling the information obtained and providing greater accuracy in aligning the short trees to a reference genome. This information is typically stored in a FASTQ file format, which we will extensively talk about in our future videos. In summary, short-tree sequencing has become an indispensable tool in modern genomics, offering a balance between throughput, accuracy, and cost-effectiveness. Despite its limitations in resolving highly repetitive or structurally complex regions of the genome, its advantages make it the go-to method for a wide range of applications, from research projects to clinical diagnostics. As technology continues to evolve, improvements in read length, accuracy, and speed will only further enhance the capabilities of short-tree sequencing platforms. If you have any questions on this topic, drop them into the comments. And if you have any suggestion on which topic you would like us to cover next, feel free to let us know.